Good afternoon. I'm Shashini Shara from Department of Microbiology, Faculty of Science, University of California. So it's a great pleasure for me to be in this event, in this grand event for International Conference on Sustainable Development 2021 as an order presenter. So today I'm going to present my undergraduate research, molecular identification of fungal species belongs to Basidiomycota in pasteurized mixed berry syrup. So reason why I choose this particular fruit syrups in order to identify the fungal genera associated with these syrups is these syrups are mainly used as raw materials in food and beverage industry in order to produce various functional beverages. That means in these syrups there are a high content of various phytochemicals and micronutrients. Then these syrups are pasteurized in order to preserve them, and during the pasteurization process, the vegetative cells of bacteria and fungi are destroyed. But there's a possibility of growing heat-resistant molds and other fungal genera due to various reasons. Many researchers have focused their attention in order to identify fungal species or fungal genera associated with pasteurized fruit-based products such as fruit puree, fruit juices, and fruit nectars, but less attention has been drawn about identifying fungal species from pasteurized fruit syrups. So this is the reason why I chose this particular research topic in order to perform my research. So this is my research aim to understand the fungal flora associated with pasteurized fruit syrups. In order to fulfill my research aim, these are my research objectives: to isolate fungi from pasteurized mixed berry syrup, to obtain pure cultures of the isolated fungi, to extract DNA for the molecular identification of the isolated fungi, and then molecular identification of the isolated fungi. So what are these mixed berry syrup? Concentrated aqueous solutions that contain sugar or sugar substitute and a flavoring agent is called as syrup. Fruit syrup is a kind of syrup which is mainly formed with the combination of fruits. And this particular mixed berry syrup is a kind of fruit syrup that is made by a combination of berries: strawberry, raspberry, blueberry, and carrot juice. So these fruit syrups are used in food and beverage industry in order to produce functional beverages such as yogurt and yogurt drinks, and also the tropical fruit syrups are used to make dessert items such as ice cream toppings, pancake, and waffle syrups. So these fruit syrups or fruit-based products are pasteurized in order to destroy non-spore-forming pathogenic bacteria and vegetative cells of fungi and their less heat-resistant spores. Generally, the pH value of these fruit-based products is low, and the pasteurization temperature that is used in order to pasteurize these products is in between 80 Celsius degrees to 100 Celsius degrees. So in this particular mixed berry syrup that I have tested have a pH value in between 3.2 to 3.6, and the commercial pasteurization temperature that was used to pasteurize is 95 Celsius degrees for 4 minutes. Although these products are pasteurized, there's a possibility of growing heat-resistant molds in these products because they have the ability to produce characteristic heat-resistant structures such as thick-walled sexual spores. And when these organisms are grown in products, they can produce off odors and slightly sour taste, gases, and mycotoxins. Other than these heat-resistant molds, there can be other fungal genera such as Aspergillus and Penicillium, as they are ubiquitous in nature and they are rapid colonizers. And these organisms can enter to the pasteurized product after the pasteurization process. That means it can be a post-pasteurization contamination. There can be various reasons for that. Sometimes there can be inadequate thermal processing, then incorrect formulations, and high moisture content. Now let's move to the methodology part. First, the isolation of fungi was done according to the International Federation of Fruit Juice Producers, IFU method number 4, heat resistant mold spore detection method. First, 50 milliliter of 35. Then these contents were mixed thoroughly or homogenized in a stomacher 400 circulator for 5 minutes. After that, the heat treatment was done at 75 Celsius degrees for 30 minutes. This step is very crucial because it activates the dormant fungal spores by inactivating the viable mycelial cells. After that, the contents were heated immediately and 50 milliliters of cooled content was distributed in 4 sterile petri dishes. Here, the malt extract agar was used as the culture media, and the prepared plates were incubated at 30 Celsius degrees up to 6 to 15 days. After this incubation period, the fungal colonies with different colony morphologies was isolated as pure cultures. After that, the second part, the molecular identification was done. So, for molecular identification, first, DNA extraction for fungi was done. So, in this process, I'm not going to tell the process in detail, but I'm going to mention only the specific regions that I added during this process. First, the fungi was processed into a fine powder by freezing it with liquid nitrogen using a microcentrifuge tube. After that, approximately 40 milligrams of this fine powder was transferred to a microcentrifuge tube and nuclei lysis solution 600 microliter was added, a vortex, and incubated at 65 Celsius degrees for 30 minutes, and it was cooled. Then RNA solution 3 microliter was added and mixed. Then it was incubated at 37 Celsius degrees for 15 minutes. After that, protein precipitation solution 250 microliter was added and it was vortex and centrifuge. After the centrifugation process, the supernatant that contained DNA was carefully transferred to another clean microcentrifuge tube, and for that, isopropanol 600 microliter was added and mixed. Then it was kept at 20 Celsius degrees for 20 minutes. After that, again, a centrifugation was done and the supernatant was decanted and the pallet was air dried. For that pallet, 70% ethanol, 250 microliter was added and centrifuged, and the ethanol was removed using a drawn faster pipette. Now, from this process, we obtained the isolated DNA. Using this isolated DNA, a PCR method was followed using ITS-1 as the forward primer and ITS-4 as the reverse primer. Then using these DNA sequences are analyzed using the bioinformatics tool Quantic Express software, and the resultant Quantic sequence with blast search on NCBI. Then the sequence with higher matching was taken as the sequence for the isolated fungi. So as the results, I could identify and isolate 2 fungi species that belong to division Basidiomycota, that means Formitopsis miliae from sample number 1 and Felinus noxious from sample number 3. So according to this slide, the figure number 1 shows you Formitopsis miliae colony after 7 days incubation at 30 Celsius degrees on malt extract agar. And this figure number 2 shows you Felinus noxious colony after 7 days incubation at 30 Celsius degrees on malt extract agar. So this research was done to understand the fungal flora associated with pasteurized fruit syrup, and the isolation of fungi was done according to the heat-resistant mold spore detection method. Because this product is pasteurized and it has been identified that heat-resistant molds are the most identified mold species that can be isolated, that can be associated with these pasteurized products. And the molecular identification was done using the universal fungal primers, ITS-1 as the forward primer and ITS-4 as the reverse primer, to amplify ITS region and 5.8 is RRM gene region, and this region is used to measure the genealogical relationships of close fungi because there's a far greater interspecific variations in this region region than 18S RRMA gene region and 25S RRMA gene region. Then the ITS region or the internal transcript spacer region is the golden standard buzzword for the identification of fungi. So, I could identify two fungal species, Formitopsis meliae and Felinus noxious, that belong to the descent Basidiomycota, and these fungi have not been proven as heat-resistant fungi according to literature. That means these fungi may have entered to the product after the pasteurization process. That means this can be a post-pasteurization contamination. There can be various reasons for these. Sometimes there can be inadequate thermal processing, incorrect formulations, or high moisture content. So, as my conclusion, fungal species such as Formitopsis meliae and Felinus noxious that are not identified as heat-resistant molds are found in pasteurized mixed berry syrup. And it is necessary to check the possible route where these fungi enter the product and whether these fungi can withstand the commercial pasteurization conditions. So this is my research, and thank you.
Okay. Thank you very much, Ms. Ishara, for your presentation. Did you change did you work with different temperatures? Yeah, temperature values?
No, sir.
So then, is there any method to get that with this? Can you work with these different temperatures here? Is it?
So here, what I did is I want to isolate fungi from this pasteurized mixed berry syrup. That's why I used that particular temperature, 75
Yes.
Celsius degrees at
Yes.
Then if we want to further clarify whether these fungi have a heat resistance in different temperatures, we can make a spore suspension using these fungi, and we can test that heat resistance in different temperatures.
Right. Okay. Thank you very much again.